It's 11 o'clock and we're in Finland, and we'll start our workshop for cell tracking today. Just to give you some links to begin with, I have gathered all the information for this workshop in our GitHub page, which I will send it to the Slack, to the chat window just now. And also we have some data sets available that you can play with after the workshop. I will also send that to the chat window there. Okay, good. So welcome everyone. My name is Joanna Pylvänäinen and I'm a postdoctoral researcher in the cell migration lab in Akademi University in Turku in Finland. And I'm working with analyzing live cell image data, and this is why tracking tools and such are very essential for us in our work. So this presentation will be like a demo setup. We will first go through a little bit how to do batch processing of tracking using TrackMate, and then we'll do a little demo about that. And then in the second part, I will introduce CellTracksColab, and then we'll do a little demo on that. And all the links and data sets and training materials are available in the links that I just sent, and those should be available also in the description of this workshop. So let's start by me sharing the screen. Here we go. And just to let you know, if you have any questions during the workshop, we have Hannah Grobe here, who is my postdoctoral researcher fellow in my group. So she will answer your questions through the chat. So I have disabled the questions and answers function in Zoom, so please use the chat instead. Okay, so let's start. So today we will discuss how we can analyze cell behavior using TrackMate and CellTracksColab. How do I minimize this? Okay. Let's see if I can change. Here we go. So I will introduce two tools that we have developed in our lab. The first one would be TrackMate. This was developed in collaboration with Jean-Yves Tidives's group in Institut Pasteur in Paris. And this is a specialized software for tracking of microscopy videos. Then in our lab, we have developed CellTracksColab, which is a platform which can be used for further analyzing the tracking results and for plotting the data. Little bit introduction for TrackMate. So it's an open source plugin for Fiji and ImageJ. It's been very popular over the years and cited hundreds of times. And it's a very user-friendly software with a step-by-step wizard to help you analyze your videos. It's very flexible. It can track different biological data, and it can use different tracking algorithms to do that. You can do customizable analysis workflows, and you can also do a little bit of visualization of the tracking results. Great thing about TrackMate is that there is an extensive documentation available, and there's an active community further developing it. CellTracksColab, on the other hand, is a cloud-based tool for track analysis. So it allows you to upload, compile and visualize your tracking results, and it's very accessible. It's suitable for all researchers, even without coding experiences. And you can simply output your track data. You can do graphs and visualizations in real time. Okay, so let's start with tracking. So When talking about tracking, we have two main types of tracking that we usually talk about. So first would be particle tracking. In particle tracking, each centroid of a particle is identified, and that is tracked then over time. In object tracking, the entire shape of the object is tracked. And this allows us to, in addition to tracking the speed or direction of the track, we can also track the changes of the shape or the intensity of the tracked object over time. Tracking is also much used in different real-life situations, like we saw a little video here on the right, where people are crossing the street and being tracked. In cell applications, cell biology applications, we might want to do tracking analysis when we want to track individual proteins in living cells, or in general, different types of cell motility or migration studies. We can track cell division over time and measure organelle movement or shape or size changes over time, and the changes in their signal. In TrackMate, the tracking principle is split in two parts, for which first is segmentation, and the second one is called cell object linking. So we're going to first briefly talk about the segmentation step. So TrackMate allows us to use different methods for object segmentation. We can use classical systems as log or doc detectors, or we can input masks or label images for TrackMate to identify our objects. We can also incorporate deep learning models or machine learning models that we have trained elsewhere to TrackMate to automatically detect our cells in the workflow. And here's some examples of what kind of images we can input to TrackMate. For example, the original image, a mask, instance segmentation, or even semantic segmentation. In the tracking context, segmentation is object differentiation for segmentation is very important. For example, here we see an example where in the raw image, we can see that we have two blobs, most likely nuclei. And in instance segmentation, we can separate these two from each other. Sometimes if a segmentation algorithm is too efficient, it might separate one object into two, which is causing false objects and over segmentation. Whereas if wrong kind of segmentation is used, we can also miss some object, causing under segmentation. And this will, of course, affect our tracking results, because in the next step, we will combine these track to each other, or objects to each other in time. The outline precision in tracking can be of secondary interest, as we are more interested in linking of the object than the actual content in some cases, but this really depends on the nature of your analysis. In TrackMate, objects can be detected in different ways, as I mentioned already, and here I have included some examples that can be used. For example, using the log detector, you can generate round detected circles, but you will miss the actual shape of the cell. You can try star disk segmentation. Here I have used the versatile nuclei model, which obviously doesn't work that well for bright field images. I have tried the threshold imager, where you set a fixed threshold for your detection. Or you can even try to use the Voronoi-Otsu segmentation, which obviously doesn't detect your cells super well. So the segmentation part is very important, and therefore in the workshop today that we're going to use already generated label images using star-artist segmentation. So the labels would look something like this on the right, and just to show that TrackMate can also read mask images here. The second step of the tracking pipeline in TrackMate is object linking. And this essentially means that we need to link objects to each other in time to be able to follow where they move. In TrackMate, there are several different trackers available, and I'm just going to show some examples here. So one example would be a lab tracker, where objects are linked using a certain search radius. So in simplicity, if we have time 1 in blue here, and the time 2 would be within a certain radius, after one time point, a link would be created. Whereas if the object would be further away than the radius, no link would be created. Another example of a very different tracker would be the overlap tracker. Here, TrackMate is using the overlap of objects to define if they belong to the same track. For example, here we have time 1 in blue and time 2 in yellow again. And if these objects overlap in time, a link would be formed. Whereas if there's no overlap, no link would be formed. TrackMate also allows us to track multiple movies simultaneously. So this is done with the TrackMate batcher. TrackMate batcher allows you to drag and drop all the images that you want to track in the first window of the user interface. And on the right side of the user interface, you set the path to a TrackMate settings files. So these are the settings that you have defined. It includes the detector you want to use. If you want to do any filtering for your objects, it includes the tracker that you have selected and any other filtering as such, if you want to use something. Then you can also set the folder where you want to save your results and what kind of results you want to export. For example, the spot table and track table, which are, by the way, essential if you want to further analyze your tracking results in CellTracksColab. And then you have the log window here where you can see what's going on and if you have any errors during a batch processing. And the output will look something like this. So everything will be exported into the same folder. You will have a spots file and tracks file. If you want to export a movie, you can do that. And everything will be neatly organized in one folder. So now it's time to go for some demos. So today, our task is to study how do these T cells migrate, or how their migration is different based on what kind of substrate they're crawling on. So we use two substrates. We have ICAM and VCAM substrates. So to do this, we will go through a short workflow. I will do a demo about this now, and I recommend that you try to do this at home later as homework, if you wish. Okay, so let's start. So I have my TrackMate open here. This way. First, we want to start by defining settings for our batch processing. To do that, we open our image that we have prepared for creating the tracking settings. And hopefully it opens. There we go. So this is what the image looks like. I mentioned before that we are using an image label detector for our images, and this is because detecting these cells, bright field cells, is really difficult. So I'll show you how the image looks like without the labels. So it would look like something like this. So these cells might be very difficult to detect using already available tools in TrackMate. Therefore, we have trained a deep learning model, Stardist, to be able to segment these cells, and we have combined those two images together. Okay, but let's start. First, let's open TrackMate. So you can find it in the plugins window, or what I usually like to do, just type what I need to the search window and click run. And the TrackMate interface should show up. It looks something like this. In the first window, you can see that how your image has been read. So if the calibrations are read correctly or not, how many time points you have, and so on. So we go next. In the second window, you will select a detector. In here, we have a long list of different types of detectors that you can use. And I recommend you to play around with those a little bit if you wish, just to understand a little bit how they work. But in this case, we choose the label image detector and click next. In the next window, you have to tell TrackMate which channel you want to segment using the label detector. And we know that our labels are in the channel 2. So therefore, we select channel 2. It's a slider. You can slide it back and forth like this. And if you're unsure how it will look like, you can always click the preview window, and you will see these nice purple lines showing up around your segmented objects. Looks pretty good to me. And we move on to click next. And here, all the frames will be detected. Next, you can do some initial thresholding based on the quality of the segmented spots. We are not interested in that at that point, so we move to next. And here, different features of the spots are calculated. So, and you can set filters on these spots. For example, if you want to filter out spots that are smaller than of a certain area, you can do that here. And you can see the result or segmentation changing interactively on the image. So we can slide this slider here and see how only larger objects are segmented or detected if we choose so. There are also different types of filters that you can use. If you have a fluorescent channel with signal, you can use intensity, for example, and so on. But the purpose of this demo, we don't need to filter anything at this point. So I'm just going to remove this filter over here and move on. In the next step, we are going to link the object in time. We have several different trackers available, and to be able to select the correct tracker, you will need to know something about your data. I know that in this data set, we don't expect cells to be splitting. So it's enough that I will use a simple lab tracker, and I'm going to use that. For example, a normal lab tracker that will support track splitting, but we don't expect that to be happening now. So I'm going to go with a simple lab tracker. Click next. In the next step, we define how the settings for the tracker. So for example, what is the maximum distance of the objects? What can be the maximum distance of two objects so that they are still linked to each other? And what is the gap closing max distance? So how much can they the gap. So if you lose an object in the gap, how far they can still be attached. And here in the third one, you can define the max frame for gap closing, which means that if you lose your object for this amount of frames, it can still be connected to the same track. So I've tested these settings and I know these are good. So I'm gonna just go on, click next. And here we can start visualizing the tracks. So here we see that all the cells that move along one track have been connected to each other with a colorful line, which we call the track. Great, looks fine. So in the following window now, again, you can set filters on these tracks, but as I mentioned before, for the purpose of this demonstration, we don't need to do that at this point. So now we're happy with our settings. Our tracking looks pretty nice. I like it. It shows that all the cells have pretty much been tracked sufficiently well. So now we have designed our tracking settings. To save this, you have this little save button on the bottom. Please click that. And it should open a window and where you can save your tracking settings. And I'm just going to save it in the same folder over here. And now we have the tracking settings saved. And we can close TrackMate and the image at this point. Great. Next step, what we want to do is that we want to process multiple videos using these settings that we just created. To do that, we open the TrackMate batcher. This is inside the TrackMate helper update site in Fiji. So if you wish to use this, you need to remember to subscribe to that update site. And we click run. So in the first window, we will drag and drop the paths to all the images that we want to track. So all the images we have here. So we have cells crawling on different substrates, ICAM and VCAM, as I described before. So this is really cool, and I really like this feature that you can just select all your images and drag and drop them on the batcher. Now be careful that there's a small bug at this point, so you can never clean your whole all the files from here. So as you can see, the first one is empty. It's because it was there before. So just go ahead and remove that. You can also, just to be sure, double check that you have correct number of files in your folder and the same number in the batcher input. Okay, next we want to generate a path to the settings that we just created using TrackMate. So to do that, we'll find the file where we have our settings and simply drag and drop it to the TrackMate settings space. Okay. In the next window, we want to tell TrackMate batcher where to save all our results. So for this, I have created a folder. And as before, I will just drag and drop the address, the folder here, and the address will show up here. To further analyze the tracking results using CellTracksColab, you will need the spots table and the track table at least. You can export all different types of files here, but we're gonna focus on the spot table, which will have all the information of each spot segmented in each frame. This will include the shape and intensity information, for example. And the track table, and that will have information about the track features such as directionality, speed, etc. If you want, you can always export also a movie here. That might be nice. For example, if you want to test and make sure that your videos look correct, it might be nice to do that as well. But I'm not gonna do that. This, no, actually, maybe we can do that. We can leave it. And then when we are done with all the settings, we just click run, and TrackMate Batcher should run without problems. And now we are finished. That was pretty quick. So now we have tracked objects in 10 different movies in a few seconds. So I think it's pretty cool. We can take a look at the movies that TrackMate generated, just to see a little bit how they look like. There are some mistakes in the track, I'm sure, but none of the tracking is always ever perfect. Okay, great. So now we have generated the spots and track files, including all the important information of our tracking experiments. And I think we can have a quick look at the chat at the moment, if we have some questions that I see there's a lot of stuff happening in the chat. So I think Hannah hopefully has been able to answer. Yeah. Great, I see all the questions have been answered. Okay, perfect, thanks Hannah. Okay, so let's move on to the second part. I will share my screen again. Yep. Okay. Good. So now we move into the track analysis part. So as we realized, we just analyzed 10 movies, but in real life, we can have hundreds of movies, hundreds of multiple condition, multiple repeats, multiple field of views. This will generate huge data sets with many parameters and multiple CSV files. And analysis of these can be very difficult sometimes. So therefore, we created CellTracksColab. So basically, it's a platform that allows you easily to compile and analyze your tracking data. Currently, we have benchmarked CellTracksColab using several different tracking tools. So I showed you TrackMate before, but CellTracksColab also supports output from different tracking software such as IC, Fiji Manual Tracker, Elastic, and Cell Profiler. These we have tested, but I'm sure there's more. And if you have a data set from your own tracking tool that you will like to test, please let us know. We are happy to test it with you. The requirements for the tracking data is quite simple. So we need few columns in the data. So we need the track ID, the X position, Y position, Z position if it's a 3D data, and then the time point. And importing all this to CellTracksColab will allow us to visualize our tracks or even migration vectors. It allows us to plot the data and calculate statistics. And I will demonstrate this in a little bit. There are currently 6 different available notebooks available in the CellTracksColab platform. So we have simple analysis notebooks. We have one dedicated to TrackMate, which can directly read TrackMate output and compile these files. And then if you're using any other software, you will need to use CellTracksColab custom notebooks. This is just because of how the column names are. So different tracking software exports different column names, for example, for X or Y position or time. So the specialized notebook has to be used to map these column names correctly. And then there's CellTracksColab Viewer, which allows you to view, share your data in an easy way. Then there's some advanced analysis notebooks, like the CellTracksColab Dimensionally Reduction Notebook. This allows you to explore deeper your data. You can find subpopulations and track different features out of those, as well as the spatial clustering analysis allows you to do spatial clustering analysis for your tracking data. And the distance to ROI notebook allows you to measure distances, for example, from your cells to a certain ROI, which could, for example, be the edge of your cell colony. CellTracksColabs can be run in 3 ways at the moment. So in Google Colab, which we will do today, it's interactive notebooks that require 0 coding. If you're uncomfortable running CellTracksColab in the Google environment, or maybe your institution doesn't allow you to do that, you can also run it locally using Google Colab or by using Jupyter notebooks. And here's the link one more time to the CellTracksColab platform in GitHub, so you can take a look at it at some point. So the CellTracksColab workflow is pretty simple. So first, it starts always with the data preparation. So you generate your tracking results, and if you're using the cloud version, you need to load your data to your Google Drive. In the next step, you compile your data, and this has to be done by using one of the 2 notebooks, the TrackMate notebook or the custom notebook. All other notebooks will use the compiled data generated using these 2 notebooks. Then you can view tracks and do filtering and smoothing of the tracks in the notebooks. And you can do further track analysis, such as plotting of conditions or extracting subpopulation from your tracking data. The requirements for CellTracksColabs to work. Requirement number 1 is file hierarchy. So all the files have to be organized in a specific way. So by conditions and by repeats. So for example, here, the condition 1 is here, and under that there's 2 directories with conditions or repeats, like repeat 1 and repeat 2. Okay. And then for the data, the minimum requirements, as I mentioned before, you need to have the ID for your track, the X coordinate, a Y coordinate, Z coordinate if it's 3D data, and the time point. So with this, you can get started on CellTracksColab. CellTracksColabs allows track visualization in different ways of the raw data, or you can let the data migrate from central middle points, or you can plot migration vectors, and I will demonstrate these in a little bit. You can do track filtering, for example, smoothing the tracks, or just look at the tracks with furthest travel distances, for example. CellTracksColab also allows you to compute a lot of additional metrics, and I have just taken a small screenshot here, and feel free to reach out to the notebooks to read more about these in detail, as maybe it's a bit boring to read through all these here. Then very cool thing about CellTracksColab is that it allows you to do quality control for your data set. So as biology usually is unbalanced. Different conditions may have different number of cells and different number of tracks, etc. So it's very important to balance your data set to capture the true biological variation. So yeah, for example, here we have our ICAM and VCAM videos. So these are tracks extracted from the videos. And we can see that in the ICAM condition, we have 457 tracks in the first repeat, 306 in the second repeat, and only 267 in the third repeat. Whereas in the VCAM videos, we have 559 tracks in the first repeat, etc. And these cannot directly be compared to each other as the data sets are of a different size. So therefore, CellTracksColab allows you to balance your data set. So basically, you pick a group that has the least number of data points or tracks, and then you do randomization of the other data sets so that eventually you will have same number of data points in each condition. You can also visualize how this randomization affects your data quality. So in the next step of the notebook, the original data and your downsample data will be compared and displayed on a heat map. And if there's high differences, you can see blue or purple, blue or green blobs. And if you see everything in yellow, the downsampling has not affected your data. And this is done for each feature, for example, condition 1, repeat 1, and for example, track duration. So if you see a green or blue box at the feature that you're interested in looking at, maybe you should acquire more data, for example, to get more accurate results. Then I briefly mentioned about the distance to ROI notebook. This notebook allows you to import your tracking data as well as an image mask, where then the distances can be computed over time to the selected ROI. In the dimension reduction notebook, the nice thing about this is that as the output of the tracking data can be very complex, as it will have many tracks, many features measured, and it will be really difficult to analyze or plot these features in a simple way. So therefore, you can generate UMaps or TSNE maps, where you can see the distribution of the highly dimensional data in 2D. And you can even use HVD scan clustering to subtract different subpopulation from these high dimensional data. You can further use the dimensionality reduction notebook to find the distribution of different clusters in your data. For example, here we can see that ICAM has a lot of clusters, like only few clusters in the a large number of clusters in the group 4, whereas or high number of tracks in the cluster 4, whereas VCAM has only few tracks in the cluster 4. We can further look into these different clusters by plotting selected features from these clusters. For example, here in the first graph, we have plotted the track mean speed per cluster for ICAM. So we can see that the tracks that are in the cluster 1 move slower than the tracks in the cluster 4. Then we can also plot these by condition. So we can take the cluster 4, which we can see that has a lot of tracks in ICAM and few in VCAM and look into their differences in more detail. Okay, so let's try CellTracksColab in practice. So CellTracksColab can be found from GitHub. The address is here. You can, easiest is to just Google it, I guess. And if you go to wiki page and scroll down, you will first see video tutorials, how to get started, etc. And if you scroll down a little bit, you will find the notebooks. Today, we're going to use the CellTracksColab notebook. To open the Colab notebook, please click this Opening Colab link. This will open the notebook. And before we start anything, we want to make a copy of this notebook to your own Google Drive. And give it a name. This is, for example, because these notebooks are updated all the time if new bugs are found. And if you save this notebook as it is for your version, for your experiment, you can always go back and see exactly what was used in that notebook. You can always open the notebook again from the GitHub repository, but it might not be the same notebook. Okay, so these notebooks are very well documented, so I really recommend you to take your time and read through all the text. Here it will describe all the steps and all the information you need. But to start actually analyzing our tracks, so first we need to prepare the Colab session to start with. So we start by installing the requirements. And this will take a little bit of time. And still a little bit of time. Yeah, perfect. Next, we want to connect this notebook to our Google Drive. And there will be several places where you have to add and agree and click. You just need to deal with it. Yeah. So now you get a text that you have mounted this notebook to your Google Drive. So now you should be able to see your files. So to use the online platform, you need to upload your tracking results to Google Drive, to your own folder. And now I can see that after mounting this notebook to my Google Drive, I can actually see that there's my drive. There is folder for this workshop. And in this folder, I will see different files that I have prepared for this workshop. So great, it works. In case you fail to see your files here, you might need to click connect to a hosted runtime option from the right top. And usually this will remove the problem. Okay. So next we load the dependencies needed for this notebook. Great. Okay. So those are just to prepare the session. Next, we will tell this notebook where the data is that we want to analyze. So now we go to the folder where we have the data. So I have prepared a folder called I2K CellTracksColab input. And in here, as I described before, the requirements of the data are met, as I have organized the data so that I have the ICAMs in 3 different repeats and VCAMs in 3 different repeats. Okay, so we can close that. And then to input the path to this folder, you right-click and select copy path. And in the folder path, you just copy-paste this path. In the next cell, you will define what kind of dimensions your data has. And I know that ours is 2D, so I've selected that. And you can select the file format. We are working with CSV files today, so we will click that. If you just want to play around, you can also use a test data set that you can upload here, but we won't do that today. And then you have to define a folder for your results. So I have created an empty folder here called CellTracksColab results. And I'm just going to right-click it, copy the path, and paste it to the results folder place. So now I play the cell and things start happening. CellTracksColab starts processing and compiling these files right now. And it's done. Now we can see what it looks like in the folder. So after this step is done, in the result folder, you should have 2 files. One is called merge spots and merge tracks, and these are your compiled track files. Okay, great. So then in the next step, you can visualize your tracks. It's not a mandatory step. You can skip it if you want to go directly to the analysis, but I will just demonstrate how this works. So you can first visualize your tracks in each field of view. So if we run the first step, we can select the file name that we want to look at. For example, one of the first ICAM files. Or if we want to look at one of the VCAM files, we can select it from the drop-down menu and the image will update. So here we can already see a little bit how the tracks would look like. In the next step, if we want to process all these field of views, all the videos that we have, you can click here. So then when you do that, all these kind of images or plots from each condition will be saved on your Google Drive. I'm going to skip it now because it will take some time, but so that you know option is there. In the next step, you can run the origin normalized plots. So for example, here we select the first ICAM movie and we can see that if we normalize the start of each track to the center of the plot, it looks something like this. And similarly, you can select any other condition to view. Okay. And similarly, you can process all the fields of view here by clicking the arrow over here. Then next, we can look a little bit further into the data and compare, visualize the whole conditions. So in the next step, you can choose which condition and which repeat you want to visualize. For example, if you want to see visually quickly if there are differences in the repeat 1 or repeat 2, you can first plot for repeat 1 of ICAM. And then select the repeat 2. And see if they look similar or not. Similarly, again, you can process all the repeats and condition combinations by clicking this arrow below. And then finally, you can create plots for each condition. So we have only 2 conditions. We have the ICAM and the VCAM. So here we first plot for ICAM. Will look something like this. And then for VCAM. It takes a bit of time. Okay. And similarly, you can process all these conditions here. Okay. Next, we can filter and smooth the tracks. To do that, we start by running the basic track metrics for filtering purposes. And then you can run this cell here, run to filter, and you will get some options here. Here you can define if you want to smooth your tracks or not. And if you want to include, for example, tracks that only have a long distance. So if I apply these filters and compare, it will show me that now I only wanted this one long track to be visualized and smoothed. If you're not happy with the results, you can always go back and readjust. Okay. Then in the next cell, you can choose which data you want to use for further analysis. So do you want to use the raw data or smooth and filtered data? We did quite a lot of filtering, so let's go with the raw data. Okay. In the part 4, you can compute a lot of additional metrics for your data, which might be necessary. I recommend you to look inside the 23 hidden cells here. We are not going to run through them now because it takes just a lot of time and we are not going to be able to visualize the results in any way. But if you want to see what kind of other additional metrics you can compute using CellTracksColab, please go through the part 4. In the part 5, we're going to do quality control. So in the first step, if you run the first cell, you can check the number of tracks in your each condition. So we can see that our smallest repeat is this one here, VCAM1. So that will be used to define how many repeats will then, how many data points we will have in the all conditions later on. Then we can compute the similarity metrics between field of views, between conditions and repeats. And here will come the heat map that I showed you already. Oh, sorry, never mind. This is the similarities by the conditions. So this part will plot denograms that will show you relationships of your different data types. So in the first one, you can visualize how closely related these data are to each other by condition, or in the second one by, so the first one is by repeat and second one is by condition. So this already hints you that the features from your data actually ICAMs are similar to each other, whereas VCAMs are similar to each other. In the next cell, you can plot available track metrics. So here again, there's a lot of documentation on what kind of plottings can be done and what kind of statistical analysis CellTracksColab provides. But let's look at the first. So in the step 6.1, you can plot your entire data set. This allows you to look at the normalized z-scores of your data. So this will just tell you a little bit about the features of the data and how different the two conditions are from each other. So here we can see that the first column is for ICAMs and second is for VCAMs. And we can see that, for example, for the track mean speed, the variations between these two different data sets is quite high. So maybe we can actually extract some information from there. So let's select that as a feature of our interest, and we can go to plotting these track parameters. So we select the ICAM and VCAM conditions. So here it's a bit tricky. You need to have the shift button down and select both. Otherwise, you will just select one. So you need to select what you want to plot. And then we can select features that we want to plot. So for example, the TrackMate metrics that we generated or imported from our tracking software, TrackMate, So we can select here the track mean speed. And for example, what would be interesting for us? Track duration. So those are quite self-explanatory. What is the mean speed of each track and what's the duration of each track? And then we can click plot selected variables. And now we have generated plots. The first plot looks a bit odd, so let's take the second one. It's a bit slow. Here we go. So for the track mean speed, now we have plotted the mean speed of each track. We can see each condition here. So we have condition repeat number 1, repeat 2, repeat 3, and all conditions together, whereas the same and for ICAM. We can also find statistics. Well, first of all, the effect size that tells you about the size of the effect. Then we have the p-value test and a corrected p-value test. See, we have 4 minutes left. So I will just show you quickly the next part. So you can do the same plotting here for your balanced data set. Remember that we were selecting the 219 tracks from each condition. And then the following step, you can see how well the downsampled data corresponds to the original data. And we can see that there's some green areas, but not that many. And then in the last part, we can plot track parameters of the balanced data set, similarly as we did for the unbalanced data set. So here we got nice results for the track mean speed. And here we got the plots of how these different track speeds, how different track speeds are in these two conditions. So what we can say here that simply the cells crawling on ICAM, one substrate, crawl faster than the substrate on the cells on the VCAM, one substrate. So this is the end of this part. So if you want to explore CellTracksColab more, you can always use the other notebooks available on the CellTracksColab platform, and always reach out to us if you need some extra help. And I'll check the chat now if we have anything urgent or yeah, looks good. Yeah, so seems like all the questions have been answered. Thanks, Hannah. One thing I forgot to show you, I will show you with the minute I have left. And so after running the notebook, all your results will be stored in the results folder that you set in the beginning. So this will have your merge tracks and spot tables, your plots. For example, here we plotted the track mean speed, so that will be automatically saved in your Google folder for further editing or directly to be used in your paper. Okay, so no SVG. What was the other? You will have the outputs in PDFs, and I think there was also CSV files. So that will be your raw data PDFs, but of course you can always convert PDFs into SVG files if needed. Okay. If there are no more questions, I want to thank everybody for joining me today. And if you have any questions or want to discuss about CellTracksColab, please don't hesitate to reach out to me. You can find my email from the GitHub repository of this training course. All right. Thank you, everybody, and see you next time.